Hello and a warm welcome to our Cenibra podcast, which we named Looking at Science, relating broadly to our product lines, looking at cells and looking at layers. My name is Christoph Enz, and in this series we engage with customers, suppliers, friends and business partners and talk about science and things around it, in a very general sense. We will meet with interesting people who talk about their work, their research and their experience with us, our technologies or our common playground in science. It can be quite specific, but we will also dive into broader aspects of scientific work and topics related to it. We're doing this not regularly, but in a very opportunistic way. So when we run into an interesting topic and conversation partner and all of us can squeeze it in, you will find something in this corner of our website. New episodes will be announced on our social media, so no need to subscribe here. If you like it, just enjoy and pop by again. And if you don't, please tell us so we can learn.
Hi and welcome everyone to today's Looking at Science podcast episode. My name is Thomas and I'm a new member of the Cenibra team. I have my background as a cell and molecular biologist, and I've spent most of my time using a wide range of imaging techniques as well as flow cytometry. Now I'm combining both qualifications as a new product specialist in the making in the field of imaging cytometry. In this next episodes, I will use the opportunity to learn more about one of our partners and the device we are distributing for them, the HoloMonitor. And today we will start with the first episode of 10 questions and continue with a more in-depth discussion about the technical background and particular applications. To do this, I invited Lisa Bodily, Head of Marketing and Communications of the Phase Holographic Imaging Team, or short PHI. Hi Lisa, it's great to have you here.
Thank you, Thomas. Thank you for having me.
So Lisa, for the beginning, I would love if you could tell us a little bit more about yourself.
Yeah, sure. So hi, I'm Lisa. I've been part of the PHI team since 2018. And I started my academic journey at the TU Dresden in Germany in molecular bioengineering and continued moving to Sweden and Lund University to dive further into molecular biology and breast cancer research. And after moving to Sweden, two other things happened as well. I developed an unfortunate addiction for Swedish coffee culture and a love for Nordic nature and long distance hiking. But at PHI, I introduce fellow scientists to our quantitative phase imaging technology and our cell-friendly live cell analysis system, the HoloMonitor. I've been working closely with our customers, our product development team, our partners, and all our in-field colleagues and global distributors. Over the years, I've transitioned from a product specialist to leading our company communications. Today, you will find me presenting at conferences and events, hosting webinars, writing articles and campaigns, and as a first, being on a podcast.
Also the first for me. Thank you, Lisa. That's really interesting. Is there something you do like in particular, or what still drives you to work in this technology field?
That's a great question. So first, like we at PHI, our PHI team, we want to empower other researchers to analyze their cell cultures without influencing them. So obtaining quantitative individual cell data and knowing what their cells are up to behind closed incubator doors. And for me, talking to our customers and being able to bring innovative technology to researchers so we can improve our cell-based research models, that is very important to me. We can reduce animal testing, we can achieve the best possible results to find answers within the field of biology. We can find treatments for diseases and also make research more efficient. So more automated, more reliable, higher reproducibility, lower costs and resources. That motivates me.
That's a fantastic ambition you have at PHI. Could you briefly introduce your company to our listeners?
So Phase Holographic Imaging, or short PHI, is a leading medtech company headquartered in Lund, Sweden, and we have also a global presence. We develop and market our non-invasive lab instruments, which researchers use to study living cells and cell models in a relevant and cell-friendly way. Our technology offers an innovative approach to cell quality evaluation. It provides detailed information on cell characteristics over time without harming the cells, a relevant limitation of traditional measurement methods that we want to avoid. And we are, as a team, actively focusing on expanding from our current preclinical market within cancer, stem cell research, and drug development to the sizable healthcare industry and the rapidly growing regenerative medicine field.
Great. Thanks for this overview. So let's take a closer look at PHI, shall we? So how would you describe the vision of your company?
Yes. So we at PHI envision transforming live cell analysis and establishing our QPI, quantitative phase imaging technology, as a standard for cell quality control in regenerative medicine, making our future cell therapies safe, affordable, and accessible for patients. To date, we have already brought many HoloMonitor systems into cell lab incubators worldwide at universities and research institutions and small to large life science companies. And to be honest, many of us biologists here in the team would have wished to have a HoloMonitor in their past research projects, me included.
Oh, yes, I can imagine. It's a great piece of technology you have there. But Lisa, before you go on and tell me more about all its benefits, I would just briefly start to share my own experience of it. So although I had it in my hands, unfortunately, I haven't tried it out myself yet. That's also the reason I would love to hear your thoughts afterwards. So my perception of the HoloMonitor right now is that it's especially developed to perform long-term and label-free assays. That means that it enables the study of cells in time-lapse experiments without disturbing them at all. And I have seen how compact it is. Therefore, there's no need for environmental control since the microscope itself fits directly into your incubator. But Lisa, since you are the expert here, what do you say? There might be listeners who have never heard about the HoloMonitor before. Did I manage to capture the spirit of it?
Yes, Thomas. That was pretty good. I can, give you another run through, and I think the most striking part about the HoloMonitor is that it works label-free. So users are able to get live cell data without having to go through the staining or the labeling process of their cells. So it's a technology only based on the optical properties of your cell sample. And that means it is also a cell-friendly, as we call it, technology. And the HoloMonitor is designed to be cell-friendly, sitting inside the incubator and working on a low-power laser. That will enable you to get quantitative data. So that, I guess, is the third point on both populations and single cells. And you will get kinetic data, so data over time, because we are also using a motorized stage where you can put multiple different plates and image in multiple different wells. So yeah, label-free, cell-friendly, quantitative.
And of course, nobody wants their cells to die within one experiment. When I first heard about the device, I also remember especially this term of non-invasive imaging. Could you elaborate on this a little further? What are the major drawbacks in observing and analyzing cells in a more traditional way, and how are you improving this by using the HoloMonitor?
All right, so let's start from the top with your questions. So quantitative phase imaging, short QPI, and let's stick with QPI for the rest. It is a long word, isn't it?
We should, yes.
So QPI is the foundation of our HoloMonitor, and it's a technology that really heralds a new era in cell quality evaluation because it provides a non-invasive detailed analysis of cell characteristics without harming the cell's integrity. And that is in contrast to conventional methods that often jeopardize cell health. So when we think about traditional ways when we want to study cells, cell cultures, let's have a look. We have fixed samples versus live samples, very common. And I guess that goes without saying, actually having a living cell sample is much more real life than a fixed sample. We have endpoint assays versus time-lapse assays. So sometimes you have a setup that only allows you to go to a first time point versus your end time point. But if you have the opportunity to actually know what's going on the whole time during your experiment, you would be able to, for example, have a look at how your drug treatments could have multiple different effects over time. Or maybe you even have a subpopulation in your cell culture that is behaving differently. So that's an interesting point when you look at endpoint versus time-lapse imaging. And as I said earlier, labeling cells versus label-free. And it's not only that you not have as many handling steps in preparing your samples, which costs time in the lab, costs resources, but you're also influencing the cells. So if you have more sensitive cells, patient samples, precious samples, stem cells, that is something that our customers value a lot, being able to not having to label their cells, but still getting the quantitative data that they're after. So I guess a major benefit of our technology, which is one of the major subtypes of QPI. Now I'm introducing another abbreviation called DHM, called digital holography or digital holographic microscopy. But I guess we're going into a separate question with that one, aren't we? So in comparison of this QPI technology to, let's say, phase contrast, we're all familiar with phase contrast microscopy, is that QPI is a quantitative method. It is digital or computer-based and an accurate method that precisely detects the cell outlines from the background by only using a low power laser. So that's in comparison to phase contrast, where you don't get a qualitative picture, you get other way around. You're not getting a quantitative picture, you're getting a qualitative picture, which means you can see but not get the data from it. So in short, QPI measures, that was one of the questions, how does it work, right? So in short, QPI measures the phase of the light wave, and how that alters when it passes through a translucent object, such as the cell. And it's a great and cell-friendly method. I hope that answered all of these questions.
Definitely. And you're right, it's just a great and cell-friendly technology. And we will definitely also meet again to tackle a separate episode to discuss some applications in more detail. However, as a first overview, what types of cell analysis and assays is a HoloMonitor well suited for?
All right, that is a very common question. And so within the research areas of cancer research, stem cell research, or in vitro drug development, I think that's the areas our customers use the HoloMonitor systems to study their cells' behavior and monitor the cell quality and the cell health. So to be honest, it's basically like a 24-hour live cell TV. You want to know what are your cells up to when you close that incubator door. Like what are these guys up to, right? So what could they be up to? They could be growing, they could be dying, they are moving around. You could measure cell migration, motility, morphology changes over time. You can run drug dose response experiments, wound healing scratch assays, cell proliferation, single cell tracking. I could go on. Maybe we keep it at that. That's already quite a lot.
And that sounds quite versatile. However, every application always needs also someone who is eager to use it. If they don't do it already, who should be interested in using your device? So which biological questions could be answered? And would you say the HoloMonitor could even represent a complementation of already existing laboratory equipment?
Yes, absolutely, Thomas. So we believe the HoloMonitor is a live cell analysis tool that fits any research lab or any cell lab. Literally, because also of this very compact size, right? We never have a lot of space in our labs, do we? So narrowing down, however, applications, I think is always quite difficult. As you said, it is a very versatile system. So you can study different facets of, everyday cell life and cell behavior. But recently I spoke to a group that was investigating the reaction of various cancer cell lines to different substances and combinations of substances over a multi-day experiment. So that's a classic, I would say, user case. You can use single cell tracking to characterize distinct morphologies and migration patterns and cell motility of, for example, stem cells. And typical questions. Well, what could be typical questions? Is this treatment affecting cell proliferation? Are there subpopulations of cells with different morphology? I just want to study my cell differentiation. I want to know, do all cells in my culture move around equally much? How does a treatment change individual cell area, volume, thickness, or irregularity? Does my treatment affect cell cycle time? Does my treatment affect cell migration, gap closure? There's so many questions that people could ask and find answers to. So I think you also asked about the workflow, was it? Yeah, workflow as well. So definitely, HoloMonitor fits into an existing workflow, and we and the application team here at PHI, we always work with our customers to fit it in. So we don't need to, pull a 360 on all the protocols that we already had. And it also fits in because the time-lapse analysis done by HoloMonitor leaves your cells completely unaffected. So you could take your cell culture vessel or plate and continue reanalyzing it with other methods. For example, you can do protein or DNA analysis or move it over to any other imaging technique, such as confocal fluorescence microscopy or any other more cell-invasive method.
After this answer, I can easily imagine that there are people who already think about how the HoloMonitor could help them to answer their specific biological questions. Therefore, we should also talk about the handling. Thinking about my time in the lab, there was always one expert for one kind of experiment or one kind of microscope. Of course, it's a big challenge that you need too many resources to really be able to master one microscope. And after months or even years working on it, you still don't understand everything. How would you describe the handling of the HoloMonitor and its software? And how long does it take to set up your own experiment, and who in the end will be able to work on it?
Yeah, I mean, I know exactly what you mean, and the frustration as a biologist of learning to handle complex new equipment, maybe run only one set of experiment then on it. So that's actually a point that our customers always give feedback on, how easy it is to learn and to use the system's hardware hands-on in the lab, as well as navigating the software. And we have customers who hands-on use it, from master students and then PhDs, postdocs, junior, senior scientists, technicians, group leaders, like anyone. And our product development team here in-house is a very dedicated bunch of people that are always working very close with the scientists, sort of in-house, us, and then in the field, and also with the feedback from our customers. So we're continuously to develop a tool that is truly designed by and for biologists. Now, setting up an experiment using the software is actually super simple and a guided process. And setting up an experiment with HoloMonitor always follows the same process. So it's a bit difficult describing it, I guess, here in a podcast and then showing it hands-on, but I will try to do my best here. So picture first that you have your HoloMonitor installed in your cell culture incubator in your cell lab. And as a next step, you seed your cells and you treat them. How are you doing it usually? So nothing of the ordinary here. And setting up an assay with HoloMonitor is usually done within a couple of minutes. So first, when you have then your cells in your vessel, you click that vessel onto the motorized stage of the HoloMonitor. Then in the software, you select which assay app you want to run and add experiment information, select which vessel you're using, add your treatment, like all the normal information. And our system does not require any special vessels or plates, by the way. So many formats and manufacturers are even included in the software as an easy template. So that goes from slides to Petri dishes up to 96 well plates. Then next, you decide on what we call the capture duration. So how long you want to image, the interval, how often you want to image, and the number of capture positions. How many positions imaging field of views do you want in each well and in total of the plate? I guess then you just start image capture and you could walk away, grab a coffee, talk to your colleagues. But if you're more like me, always very curious to look and see if everything worked well, you could stay and look at the instantly presented results, pictures, and in the guided assay, also in the graphs. And of course, at the end, when everything is finished, you could go and dig deeper into the results and make a more extensive analysis.
And to be honest, we will all do it. Let's
yes, right? We're all usually very sneaking back and like, is this working?
But you're right, that sounds quite easy and user-friendly indeed. But in terms of data presentation, let's pretend I already bought my own HoloMonitor, if I had some money, and got some really interesting and beautiful data. If I now want to publish my results, how is my data described in the end?
So all data, and that is also images and timeless videos and all the raw, what we call like the numerical data, tables, graphs, all that is exportable for post-processing, of course. So Excel and CSV and all the different common image and video formats. Working with live cell videos, to be honest, is always giving me a lot of joy because it's very colorful. So as we're working with digital holography, all cell images are computer constructed and they can be shown in a grayscale, but you can also add artificial colors to the images to make science even more fun and colorful without using labels. That's always nice to say at the end, without using labels. So yeah, you can make your images and videos more or less publication ready in the software. And once you've taken the images with HoloMonitor, your data is there. So it's stored within the image and you can analyze that taken experiment whenever and wherever you want, which became very crucial to many labs during the pandemic as they physically could not enter their labs to do more experiments, but then instead could do their data analysis and reanalysis from home. The best part, to be honest, is because we all have the data, right? So the best part is the reanalysis ability of HoloMonitor. So you can reanalyze a taken experiment image series with another app in the software to get multiple results from the same experiment and the same sample. So, Thomas, you could start with a cell proliferation assay, but also see in maybe the time lapse that your cell motility is different. Or you look at individual cells and then maybe their cell division is different or a subpopulation is showing an interesting morphology shift after a few hours, right? You can really go digging into your data.
Beautiful. And that's so a huge advantage, being able to reanalyze your data from time to time. So, Lisa, reaching the end of this first episode, let's also talk briefly about some positive examples and the successful use of the HoloMonitor. There are close to 300 peer-reviewed articles featuring the HoloMonitor so far. Can you describe some specific research areas where it has already provided valuable insights? Are there even exciting new applications or research areas the HoloMonitor is being applied to which you haven't thought of before?
Oh my, yeah, there are new articles coming all the time, and specifically in cancer research. So, for example, studying a metastatic from a non-metastatic cell line in terms of their migration pattern or EMT, epithelial-mesenchymal transition, or testing treatments, substances, or investigating the effect of a knockdown for studying cell death or cytotoxicity in drug development. These are all like really spot-on cases for HoloMonitor. For example, was another story I just had in front of me where HoloMonitor was used to study the morphology change with treatment, including an individual cell's average area, was it optical thickness and optical volume. And to be honest, that would have been a great addition to my past research project when I tried to understand better how ER-positive breast cancer cells gain resistant to treatment.
Yeah,
that
I
would
can
have been
imagine.
great. Other questions that are great to answer or were answered already were how are cells responding to outside stimuli, for example, normoxia and hypoxia? How are endothelial cells responding to flow in a microfluidic setup? HoloMonitor was also used for studying cell movement, invasion, and morphology of cells grown in a 3D matrigel environment. And to be honest, with all this now in the pocket, you could say, this is something where PHI is going. And I just want to mention that again, we are now focusing to further developing our technology for cell-friendly cell quality control in the regenerative medicine cell manufacturing process, trying to address that central piece for standardized cell quality control to make cell-based therapies accessible, affordable, and patient-safe. And that highlights the technology's capacity generating this vast amount of data for precise cell counting, viability checks, or assessing individual cell morphology that may be predictive of cell quality and ensuring the quality of a cell therapy or therapeutic product. And I particularly love seeing HoloMonitor's use grow over the years and people starting to adapt label-free QPI imaging with new setups, application areas, cell lines, like all the new things happening in research. And it is a game-changing technology, and I'm very much looking forward to many more researchers giving it a try.
That makes already two of us. Thank you so much for being here today. I'm already excited about meeting you again to talk more about the technical background and to dive deeper into some particular applications. So, Lisa, in the end, how can people reach out to you?
To contact our PHI team, you can write to us at info@phiab.com or visit our website. So that would be also phiab.com.
Excellent. Thank you so much, Lisa, for being here today and for recording this episode with me. And thanks also to all of our listeners for joining us. And you can hear us the next time in our Looking at Cells podcast. In the meantime, and if you have any questions, feel free to reach out to one of our specialists and visit our website at cenibra.de.
This was Cenibra's Looking at Science podcast. Thanks for listening, and for any feedback, please send us a note. And if you follow us on social media, you'll be alerted about the next issue. All the best, and talk soon.